Hello, everyone, and welcome to today's webinar titled The Fungus Among Us, Detecting Candida auris Colonization in Yeast Bloodstream Infections. This webinar is part of the 10th Annual Microbiology Virtual Event Series brought to you by LabRoots and is sponsored by Diasorin. For more information on our sponsor, visit diasorin.com. I am Cassie Saltman of LabRoots, and I'll be your moderator for today's webinar. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by communicating with other attendees using our group chat feature found on your screen. If you have trouble seeing or hearing the presentation, click on the Help button located in the navigation bar. Now let's get started. I now present today's speakers, Dr. Sarah Elliott and Dr. Sherry Dunbar. For a complete biography on our speakers, please visit the presenters window. Dr. Elliott, you may now begin your portion of the presentation.
Thank you, Cassie, and thank you everyone for joining us today on our session about fungal infections. The learning objectives for this session are listed here, and they can also be found on the LabRoots website. So diving right in with Candida auris and colonization. I'd like to begin by reviewing some what I think of as interesting facts about Candida auris that you may or may not be aware of. Candida auris was only discovered fairly recently, with the first case report published only in 2009, but since that time, this organism has spread all across the world. Candida auris is considered a significant public health threat for many reasons, one of these being that Candida auris has the ability to cause invasive bloodstream infections, which can lead to death, and particularly affects those in the hospital and nursing home patients that have serious underlying medical condition. So unfortunately, Candida auris seems to impact our most vulnerable patients the most. The mortality rate associated with Candida auris infections is quite significant, and it is estimated that more than 1 of 3 patients that develop an invasive infection will unfortunately die. Candida auris is able to colonize and persist on environmental surfaces and patients, and this organism spreads very easily from patient to patient and within the environment, and so it is a source of hospital-acquired infections. Antifungal medications commonly used to treat Candida infections are often ineffective against Candida auris due to its multi-drug resistant nature. Some isolates are resistant to all 3 known classes of antifungal drugs, making treatment of these types of infections challenging. It can also be challenging to identify Candida auris in the laboratory using certain methodologies, including biochemical and phenotypic methods, and these methods can misidentify Candida auris as other closely related organisms. This misidentification can lead to either inappropriate patient therapy in the context of invasive infection or missed opportunities for the implementation of infection control measures in the context of colonization. This is a phylogenetic tree of Candida auris and other closely related organisms in the Candida genus. Candida is a broad and polyphyletic genus with over 500 species, and many are markedly different from each other. Candida auris shares the genus Candida with common commensals such as Candida albicans and Candida glabrata. Now you might notice an asterisk here by Candida glabrata. This is because this organism, like many other fungal organisms, have recently undergone some changes in nomenclature, though for the purposes of this talk, I will refer to it still as Candida glabrata. So many of the organisms in the Candida genus can be quite divergent from each other. And it has been reported that Candida albicans is as genetically divergent from Candida glabrata as humans are from zebrafish. However, there are many closely related organisms within the Candida genus. So highlighted towards the bottom of the figure here, we can see Candida auris, and we also notice that there are several clades associated with Candida auris. This figure outlines clades 1 through 5, although there has been more recently a clade 6 that has been also discovered. So there are currently 6 known clades of Candida auris, although it's important to keep in mind that really only clades 1 through 4 are considered the major clades of Candida auris, and these are the clades that really have spread around the globe and have been detected in many countries, including here in the US. I also highlight this region here because using certain phenotypic and biochemical methods for identification, Candida auris can be misidentified as other closely related Candida species, including Candida hemolonii and Candida pseudohemolonii here. And we'll talk a little bit more about different methods for the detection of Candida auris later in this section. So to put Candida auris in a little bit more historical context, it was only back in 2016 that the CDC released a clinical alert to healthcare facilities warning of the international emergence of Candida auris infections, which were associated with high mortality rates and multi-drug resistance. And concerningly, this organism was found to spread very easily between patients. This emerging fungal pathogen was also starting to gain some national attention, and even the New York Times published an article titled "A Mysterious Infection Spanning the Globe in a Climate of Secrecy" back in 2019, and this article was focused on how the rise of Candida auris in the US was very concerning. And unfortunately, I would say the COVID-19 pandemic that started very early in 2020 definitely diverted attention away from this emerging pathogen, and Candida auris had certainly continued to spread throughout the US as well as other countries in the world during this time. The figure on the right is some of the earliest data on Candida auris cases reported in the US, and the trend of increasing case numbers has only continued until today. So Candida auris, as I mentioned, is considered a significant threat to healthcare. So to summarize, it is really for these three main reasons. One, Candida auris is often multi-drug resistant, and some isolates have been identified that are resistant to all three of the major drug classes of antifungals. So these infections can be very challenging to treat. It can also be challenging to identify Candida auris in the laboratory, specifically when using phenotypic or biochemical methods. And this Candida auris can often be misidentified as other closely related organisms. And finally, we know that Candida auris has caused outbreaks in the healthcare setting, so that's very concerning. So it's very critical to quickly identify Candida auris colonization so that healthcare facilities can implement their infection control measures to limit the spread of this organism. Looking at the epidemiology of Candida auris, it is well appreciated that this organism is now present all over the world. Here in the US, we consider Candida auris to be one of the top 18 antimicrobial resistant threats. And as I mentioned, Candida auris cases have been reported in many countries throughout the world. I think we're at about 50 countries at this point, including the US. And we spend a significant amount of money in medical costs treating Candida auris. And really, some of the major concern about this organism comes from the fact that 90% of clinical isolates of Candida auris are resistant to at least one drug class, usually the azoles, and 30% of Candida auris isolates are resistant to up to 2 or more drug classes. So again, it's very challenging to treat these infections. 50% of the clinical cases are bloodstream infections, which are unfortunately associated with the most significant mortality rates. There is a between 30 to 60% in-hospital mortality rate associated with Candida auris bloodstream infections. Looking at the cases of Candida auris that are reported here in the US, here we have data from the CDC. The most recent data available is from the calendar year of 2022, and it is broken down by the number of cases reported by state, and those are all shown here on this map of the US. In the year 2022, there were reported over 2,300 clinical cases, meaning invasive infections, with Candida auris, and we had over 5,700 reported cases of colonization. Now, this is a pretty significant increase if we look even just a couple of years back. So if we compare the data reported in 2019 with the most recent data from 2022, we see an over 200% increase in the number of clinical cases that were reported and an over 300% increase in the number of colonization cases that were reported. And there's no indication that these rates are expected to slow down. Now, the overall prevalence of Candida auris ranges quite broadly from 5 to 30% of all of the Candida cases that are reported from certain states. And really, this range really just depends on the prevalence of the number of cases reported of Candida auris. And unfortunately, Pan-resistant outbreaks of Candida auris have been identified in the US, meaning isolates that are resistant to all three of the major classes of antifungal drugs. And this occurred starting back in 2021. So these pan-resistant strains are present here in the US. So I'd like to talk a little bit about why testing for Candida auris colonization is so important. Now, it's important to remember that colonized patients are by definition asymptomatic. Colonization in and of itself does not cause any symptoms. So unless we're testing for colonization, really, we wouldn't be able to identify these patients. And it's important because colonized individuals have a risk of spreading Candida auris to other vulnerable patients. And we know that patients can be persistently colonized with Candida auris for months at a time, if not longer. Colonization itself is also a known risk factor for conversion to invasive infection. And it is estimated that about 10% of individuals that are colonized with Candida auris will at some point develop an invasive infection. And remember that this is associated potentially with a quite high mortality rate, where 1 in 3 patients that develop an invasive bloodstream infection will unfortunately succumb to that infection. And then finally, number 3, we don't have any currently known decolonization procedures that are effective against Candida auris. So we can certainly identify patients that are colonized with Candida auris, and we can use contact precautions and infection control measures to limit the spread in facilities, but we really can't decolonize these patients at this time. So Candida auris is a very hardy organism, and it's very challenging to eliminate this organism once it is established in a certain facility. So over time, there have been many different specimen types that have been considered for detecting patients that are colonized with Candida auris. And the most sensitive and cost-effective specimen type has been determined to be the bilateral axilla or armpit and groin swab, usually collected in a single swab. Now, certainly Candida auris can colonize other areas of the body, including the nares or the nose, the mouth, the external ear canals, the hands and finger webs. However, many studies have shown that the axilla and groin regions are the most consistently colonized sites on the body, and therefore it is the most sensitive specimen type to consider. So the CDC has a lot of great resources for all sorts of clinical testing, but very specifically for Candida auris colonization testing as well. So the CDC has a nice list of screening recommendations that could be considered, essentially highlighting those patients that are at the highest risk for colonization. And they can be broken down into really 3 main cohorts of patients. So the first being patients with an epidemiologic link to another patient or resident that is infected or colonized with Candida auris. So basically, patients that are sharing the same room or unit or other care area as a patient that has Candida auris, receiving care from the same healthcare personnel during the same time as a person with Candida auris, and also being exposed to common mobile medical equipment is also a risk factor. In the second group, this is with patients with current or previous healthcare encounters or stays at facilities with the below qualities. So patients that have had a healthcare stay in a facility with currently either suspected or confirmed Candida auris transmission, high acuity post-acute care facilities, or patients that have had a stay in a facility located outside the US, or this is a newer part of the recommendations, patients that have had a healthcare stay or encounter in a part of the country, meaning the US, with a high burden of Candida auris. And finally, cohort 3, these are the patients that have risk factors for acquiring Candida auris. So basically our most vulnerable patients. And these include patients on mechanical ventilation, those with indwelling medical devices, those that are receiving high acuity medical care, those that have frequent or longer healthcare stays, especially at high risk facilities. And finally, there have been many studies that have shown a link between colonization with Candida auris and also colonization with other multi-drug resistant organisms. So there is a strong co-colonization with Candida auris and other, say, carbapenemase producing organisms. So there are also multiple different screening strategies that can be considered. Essentially, when would a facility screen patients that might meet the risk factors that I outlined on the previous slide. So one strategy would be to screen certain patients upon admission, and this would be useful to identify potentially new cases of Candida auris that would be entering the facility, and this would allow for the implementation of appropriate infection prevention measures. And this would also serve to identify other facilities that may have ongoing Candida auris transmission that they may or may not be aware of. It would also be possible to screen a patient during a healthcare stay, and this might be performed in order to identify potentially new cases resulting from intrafacility transmission, so more part of a point prevalence survey, or just to assess the effectiveness of your own infection control measures. And finally, patients may be tested upon discharge or transfer to another facility. And this would be to identify patients or residents that might need infection prevention and control precautions at that receiving facility. So now I would like to just very briefly discuss the different types of methodologies that are available for the detection of Candida auris today. So of course we have phenotypic identification or culture-based methods. Candida auris grows very readily, of course. However, Candida auris does display some fairly nonspecific phenotypical features that can make it difficult for a conclusive identification based on culture alone. So the CDC does not recommend using culture as the only method of identification, but it can aid in that species identification. So essentially, recovered colonies require further analysis for that species level identification, and this is usually accomplished through using multi-TOF mass spec, which I'll talk about on the next slide. Biochemical identification is a method that is commonly used in laboratories to identify microorganisms to the species level. However, in the particular case of Candida auris, biochemical identification is not recommended. And this is essentially because there have been many reported studies that have found that most of these biochemical platforms have misidentified Candida auris as other closely related organisms. And for this reason, biochemical identification is really not recommended in the case of Candida auris. So proteomic identification or multi-TOF mass spec is another potential method for the detection of Candida auris. And this would, of course, be coupled with culture as the input. Multi-TOF mass spec is a very high throughput and accurate method for the identification of Candida auris. This method can differentiate Candida auris from other Candida species and other closely related organisms, as long as the lab is using the most updated reference databases. So really, this is a good method for the detection of Candida auris. The only consideration is the time it takes to get that result. So certainly once the process is started on the mass spec instrument, that's a relatively fast process. However, we do have to wait for that isolated culture as the input for this analysis. And this can take up to 72 hours, if not a little bit longer, depending on whether the specific lab is using some kind of subculture or what that protocol looks like in order to get that colony ready for mass spec analysis. And so really, this is the main consideration. So it kind of depends on why the facility is screening for Candida auris colonization and when they are performing this testing. So for example, if a facility is screening patients upon admission, typically those patients would be placed in isolation and under contact precautions while waiting for that laboratory result, in order to limit the potential spread of Candida auris in that facility. And in instances where those patients are not colonized with Candida auris, then they're sitting in these very expensive isolation rooms needlessly. And so perhaps a faster result would be more beneficial. So the final methodology that I will discuss involves molecular identification or PCR-based methods. I think that it is widely appreciated that molecular identification allows for fast and accurate identification of most microorganisms. And PCR itself has some direct benefits compared to culture. PCR is usually associated with an increase in sensitivity and specificity. And there are also some operational efficiencies that can be beneficial, particularly in the case of identifying patients that may be colonized with Candida auris, because really speed is the name of the game in order to effectively manage infection prevention and control. So I would like to mention that Diasorin has very recently received a De Novo grant for our Simplexa Candida auris direct assay, which is a first of its kind fungal PCR assay, which is aimed at the detection of Candida auris colonization. And so now I will spend the next few minutes talking a little more specifically about Candida, the Simplexa C. auris direct assay. So here is the intended use. I will let you take a look at that. I won't read the entire thing. This assay is meant to be used in correlation with the Liaison MDX instrument. And here is the consumable. This is called the direct amplification disc. And so looking at the intended use, this Candida auris direct assay is a real-time PCR assay for the qualitative detection of Candida auris DNA in bilateral composite axilla groin swabs from patients that are suspected of Candida auris colonization. Below in this table is a description of all of the different components that come with the test kit. So, of course, the Simplexa Candida auris direct kit would come with 24 pre-aliquoted reaction mix vials, as well as a sample prep kit. And I will describe more about what this kit is and how it's used in a few slides. And then we also do offer a Candida auris positive control as well. The acceptable specimen type for this assay is the composite bilateral axilla groin swab. These specimens, once collected, can be stored for up to 48 hours at room temperature or up to 7 days refrigerated at 2 to 8 degrees Celsius. After those 7 days refrigerated, the specimens should be frozen at -70 and in accordance with the transport media instructions for use. So the transport media types that are included with this for use with this assay include the eSwab liquid-based collection and transport system with either 1 ml of BD eSwab or Copan eSwab with liquid amines or equivalent. So very briefly, I'll just walk through the overall workflow for this assay. Essentially, you would just take your sample, which would have your 1 ml of liquid amines, and you would take 50 microliters of that sample, and you simply add it to the sample processing solution tube that would come in its own box. You just pipette mix up and down to mix it, and then you could even use that same pipette to load your specimen onto the disc. And then you would use a new tip, and you would add in 50 microliters of the reaction mix and add that to the disc. And then you would simply seal the foil, place the disc on the instrument, and press run. And the run time for this assay is 105 minutes. So you'll have your results in less than 2 hours. So now I'd like to just briefly go over some of the analytical performance data from the Simplexa C. auris direct assay. So of course, we performed analytical sensitivity studies, and we used 2 different clades, clade 1 and clade 4. And we found that the limit of detection for clade 1 was 127 CFU per mil, and the limit of detection for clade 4 was 260 CFU per mil. These are very similar findings. And if we do some quick math, then both of these limits of detection work out to be single-digit copy CFU per reaction. So this is a very sensitive test. One note, clades 1 and 4 were chosen because these are the most common clades that circulate here in the US. We also performed some analytical reactivity studies, and this is very important considering how many different clades have been identified of Candida auris. And so we found 100% detection of clades 1 through 6 using a combination of either direct wet testing or in silico analysis. We also performed microbial inhibition studies. And essentially, these type of studies are aimed to determine whether the assay can still detect Candida auris in the presence of other closely related organisms or those that may be also expected to be present in the same anatomical sites of the axilla and groin region. So we tested 34 different organisms, and we found no microbial interference was detected. The sample storage stability claims are listed here. Just very briefly, these specimens can be stored at room temperature for up to 48 hours. They can be stored refrigerated for up to 7 days, and then after that time, they should be frozen at -70. We performed analytical specificity testing as well, and we found 100% analytical specificity, meaning zero cross-reactivity was found with a whole list of different closely related organisms, and again, those organisms that would be expected to be found in the axilla-groin regions as well. And finally, we performed interfering substances testing, where we tested 36 different potentially interfering substances and determined whether Candida auris could still be detected. And so Candida auris was detected in the presence of all interference tested. Now, there is a little asterisk here, and this is because we did observe some interference with high levels of one deodorant cream and one antimicrobial. However, Candida auris detection was rescued at lower concentrations of these substances. Now, we don't anticipate these substances will pose any significant impact on assay performance, and this is because in our clinical study, in which we tested over 2,000 specimens, we found an invalid rate of less than 1%. So diving into the clinical performance, our clinical study consisted of 6 separate sites, 5 of which were here in the US and 1 was based in Italy. We tested over 2,000 specimens, and over 1,900 of those were collected prospectively, and we also included 90 pre-selected samples. We found a 2.8% Candida auris prevalence rate in our prospective study population, and our clinical comparator was culture followed by multi-toph mass spec identification. This table summarizes the clinical performance of the Simplexa Candida auris direct assay. So our positive percent agreement that we found compared to our clinical comparator of culture and multi-toph mass spec was 94.1%, and our negative percent agreement was 98.8%. So overall, the clinical performance of the Simplexa C auris direct assay is quite excellent. And finally, I will briefly summarize my section here. Candida auris is a global source of antimicrobial resistant infections, and cases have been reported across the world. The World Health Organization and the CDC have both listed Candida auris as a critical and urgent antimicrobial resistant threat. Candida auris spreads quite quickly and easily in the healthcare setting, and it can lead to outbreaks, particularly in our most vulnerable patient populations. Identification of patients that are suspected of Candida auris colonization is essential to support infection control measures and help facilities implement strategies against transmission of the organism. The Simplexa Candida auris Direct is the first De Novo FDA-granted PCR assay and offers an easy-to-use workflow for fast results to aid in the prevention and control of Candida auris colonization in the healthcare setting. That concludes my portion of the talk, and I will yield the floor to Dr. Sherry Dunbar.
Hey, thanks, Sarah. Hello, everyone. So I have a tough act to follow, but I am going to pick up where she left off and now focused on yeast bloodstream infections. So I'd like to start by taking you through the patient journey to get to a diagnosis of a bloodstream infection and sepsis. It's based on the general principles of clinical management for bloodstream infections. And you see there's a lot of steps that we have to get through to get from the beginning to the end. And they can vary depending on the individual patient characteristics, protocols in place at the particular institution, and also looking at local epidemiology. Now, bloodstream infection is simply the presence of a viable microorganism in the bloodstream, and this can lead to sepsis. Sepsis is a life-threatening condition that occurs when the body's immune system overreacts to the bloodstream infection. It causes severe inflammatory response, blood clots, and leaky blood vessels throughout the body. So we definitely want to find out about a bloodstream infection immediately and start to treat that patient so that we can prevent it from progressing to sepsis. So what is known is that early recognition and intervention are critical to improving the outcomes for these patients. Each hour of delay in the diagnosis and treatment of these infections leads to an increase in mortality, as much as 8 to 9% for each hour in some studies. So let's begin. So first, we have symptom recognition and presentation. The patient experiences symptoms such as fever, chills, rapid heartbeat, rapid breathing, maybe confusion, other signs of an infection. So the patient may present to the emergency room or an urgent care facility, or may even be referred by their primary care physician's office. So the next step is the initial assessment and triage. So the healthcare provider is going to do a thorough assessment of the patient's vital signs. They're going to get a complete medical history and assess the clinical symptoms, and then determine the severity of the illness. If a patient is suspected to have a bloodstream infection or sepsis, they're going to be prioritized for immediate evaluation and treatment. So the next step in this journey is, of course, the diagnostic testing. Diagnostic tests are crucial to confirm the presence of a bloodstream infection and also to identify the causative pathogen. Common tests include, of course, blood cultures, also doing a complete blood count or CBC with differential to determine, infective state, white cell count, types of white cells, also looking at lactate levels. And then we're going to measure some inflammatory markers such as C-reactive protein and procalcitonin. Other imaging studies may be conducted at this time, such as chest X-rays or a CT scan, to try to identify the potential source of infection. So typically, people don't just all of a sudden catch an organism in their bloodstream. It usually comes from somewhere. So another infection, and then it progresses and gets into the bloodstream and leads to bloodstream infection and sepsis. So blood culture collection. Blood cultures are drawn to try to isolate and identify the specific organism that's causing the infection. Multiple sets of blood cultures should be collected over a specified period of time to increase the sensitivity of detecting. Laboratory analysis involves incubating the samples to allow for bacterial or fungal growth. Let's talk about the blood culture process in a little bit more detail. So the blood for culture is drawn. And typically, the blood culture bottles are going to be inoculated right there on the floor, quickly as possible. Pre-analytical steps include gathering the supplies, labeling the bottles, performing hand hygiene, and donning the appropriate PPE, and selecting the site for venipuncture. A tourniquet's applied, the site's disinfected. Also, the bottle cap or septum is going to be disinfected. And samples are then drawn from each site, at least 2 separate sites, in rapid succession. Now, patients may have an indwelling medical device, such as a catheter, and it's really convenient to draw from there. But if one sample is going to be drawn from that port, at least one additional sample should be collected from a peripheral venipuncture site, because otherwise it would be difficult to tell whether it's truly a bloodstream infection or just colonization of that device. The inoculated bottles are transported to laboratory and put into a continuous monitoring blood culture system that's going to provide optimal conditions for microbial growth. Now, these systems automatically sense the production of carbon dioxide as an indicator of microbial growth. The BacT-Alert, for example, senses changes in carbon dioxide concentration through a color change in a pH-sensitive device in the base of the bottle. The BD BacTech uses a fluorescence sensing technique to detect carbon dioxide. And the VersaTrek is monitoring pressure changes in the bottle head space as a growth indicator. So then when the system flags positive, the laboratory will perform additional testing to identify the microorganism, which we're going to talk about in a few slides from now. But next, the next step, step 5, is empiric antibiotic therapy. So for suspected sepsis, empiric therapy is initiated promptly. And at this time, we don't really know what's causing the infection. We're going to use a broad spectrum antibiotic that's going to cover the likely pathogens that may be present while we're awaiting the culture results. The choice of the antibiotic depends on the patient's clinical condition, the site or suspected site of infection, and also looking at local antimicrobial resistance patterns, as well as recent antibiotic exposure. So now on to the lab. The microbial identification and susceptibility testing that's going to be performed in the microbiology lab. So once the blood cultures yield positive results, the organism is identified using a variety of different techniques. First of all is the gram stain, which allows us to visualize the morphology of the pathogen under the microscope, and it gives us a pretty good idea of what direction we're going. Is it a gram-positive cocci? Is it in pairs or chains? Is it in clusters? Is it a gram-negative rod? This is all giving us ideas of what might be causing the infection. We're going to perform biochemical tests and use automated microbial identification systems. Antimicrobial susceptibility testing is then conducted to determine the most effective antibiotics so that we can switch to a targeted therapy. So the identification of the organism from the positive blood culture. There's a variety of different technologies that can be used for identification. One example is shown here in the micrograph of the peptide nucleic acid fluorescent in situ hybridization, or PNA FISH. And so you can visualize the organism, in the blood culture sample. However, this is limited to just a few select pathogens such as Staphylococcus aureus, Enterococcus faecalis, some gram-negative organisms, and then several of the Candida species. There's also multiplex PCR and microarray technology shown here down in the bottom in our own Diasorin gene system. This allows rapid identification based on the nucleic acids present of the organisms that have grown in that blood culture bottle. This can allow for identification of the organism as well as some antimicrobial resistance genes in about 2 hours. And it's been shown in the literature that implementing a rapid test such as this one is associated with a decreased mortality when it's also used in conjunction with an antimicrobial stewardship program. So if we can get the answer, the lab can get the answer in 2 hours, they can communicate that immediately to the ASP program, and then they can get down there on the floor or call and contact the healthcare provider to let them know what the organism is and get them on appropriate therapy more quickly. And so the faster that we can get the patient diagnosed and treated, the less risk there is for that mortality rate that we talked about a few slides ago. There's also MALDI-TOF and micro mass spectrometry, MALDI-TOF MS. This has been FDA cleared for identification from isolates once they've been subcultured onto solid media. And then there's also conventional biochemical testing. The old manual API, which many of us know and remember, where we have these little bubbles which contain different culture media and different color indicators, which show us enzymatic activity for certain enzymes that are present, and also different sources that the organism is able to use to utilize different substrates for growth. And so by looking at the color and the pattern in these strips, we can determine the identification of the organism by unique biochemical reactions. Of course, this is also done automated, such as the Vitek 2, which can continuously monitor and read the biochemical reactions inoculated in a little cartridge that goes in there, a little slide with wells, and then tells us what that organism is. So the results are then sent to the healthcare provider, which includes the identity of the organism and also antimicrobial susceptibility if it's been requested. Now, the interpretation of the positive blood culture result does not rely solely on what the organism is that's isolated. There are certain things that we know from years of experience that tell us whether it's likely to be a true pathogen or possibly a contaminant. And also we look at the number of the bottles that come up positive with the same organism. So for example, some isolates are never contaminants. If that blood culture comes up positive with Brucella, Francisella tularensis, Histoplasma capsulatum, this is easy to interpret. If that's present, it's bad. It's always a pathogen. Isolates such as Staph aureus, Streptococcus pneumoniae, E. coli, and other members from the order Enterobacterales, Pseudomonas aeruginosa and Candida albicans are almost always considered pathogens, but in some rare cases, it can represent contaminants. And then other species like Cutibacterium, Bacillus, except for Bacillus anthracis, and most Corynebacterium typically indicate contamination because these are common skin flora and also present in the environment. This is where looking at the number of positive blood cultures becomes important. So if a coagulase negative staph grew in 1 of 4 bottles, then maybe I'm going to think it's probably a contaminant. But if it grew in more than one set of blood culture bottles, or if it grew in all the bottles, then we're going to consider it probably a pathogen. And so you take those kinds of things into consideration after you report the results and the doctor is considering whether they think it's a true pathogen or not. So now the next step is antimicrobial susceptibility testing. So there's a lot of different methods for AST. Most of the techniques, a lot of them are phenotypic. So actually looking for the inhibition of the growth of that organism in the presence of the antibiotic. And so there's different methods like broth microdilution shown up here in this microtiter plate. So twofold dilutions of the antimicrobial agent is plated in the microtiter plate in a broth medium, and then the isolates are added and the plates are incubated. The lowest concentration that inhibits visible growth is considered the minimum inhibitory concentration, or MIC. So if you look at this little red box over here, you can see the dilutions, and we get to the third dilution, and that is the last one that inhibits growth. So that is the MIC for that organism and that antibiotic. There's also disc diffusion, or the Kirby-Bauer plate, where a standardized bacterial suspension is inoculated onto the plate, and then antibiotic discs are placed on top of the surface. The plates are incubated, and then we measure the zone of inhibition to determine whether the organism is susceptible and it inhibits growth in a wide zone around that disc, or if it's intermediate and there's partial inhibition of growth, or if it's resistant, in which case, like this little disc on the right-hand side here, where the growth of the organism was not inhibited at all. There's also the gradient diffusion, which is the E-test method. And in this case, a predefined strip that contains a gradient of the antimicrobial agent is placed onto the agar plate that's been inoculated with the bacterial isolate. And then you look at the intersection of the elliptical zone of inhibition with the E-test strip, and you can't see it here, but there are numbers on these strips, and where it intersects indicates the MIC. There's also automated systems such as Vitek2, the BD Phoenix, shown here, or the microscan, where there's pre-prepared panels of antimicrobial agents in different concentrations. The isolates are then inoculated onto the panel, and then the MICs are read automatically based on the growth in each well. There's also fully automated rapid AST systems such as the Accelerate Phenotest shown down here, and it uses time-lapse imaging of bacteria under dark field microscopy and can provide a result in about 7 hours. There's also the Alfred 60 AST, which uses light scattering to detect bacterial growth in culture broth within about 3.5 hours. There are also some recent advances in direct rapid AST from blood cultures. It goes along the knowledge that most positive blood cultures are monomicrobial. That is, there's typically only one organism in that blood culture. And so advancements have been made in the use of the positive culture broth for disc diffusion or automated AST, both in Europe with the UCAS RAST 2019, as well as CLSI 2022. So for UCAS, the broth is directly inoculated onto agar and grown about 8 hours until it's confluent. And the results are interpreted at 4, 6, and 8 hours using specific breakpoints. For CLSI, published guidelines for rapid disc diffusion in the Enterobacterales follows a 16 to 18 hour incubation and uses the published M100 breakpoints. There are also genotypic methods. Molecular methods such as PCR, microarray hybridization, and DNA sequencing can be used for rapid detection of antimicrobial resistance genes. And this can provide really valuable information about mechanisms that may be present in that organism for antibiotic resistance. It's not a phenotypic test, but I can tell you if I have a Staph aureus with a MEK gene, I'm probably not going to treat that patient with methicillin. We're just going to play it safe in all cases in these types of very severe invasive infections. So all of these results, as they come out, are sent to the healthcare provider, which includes the identity of the organism and the susceptibility. Concurrent with all of this, in parallel, efforts are being made to identify the source of the infection. And this may involve further imaging studies, doing ultrasound, CT, to try to localize and characterize the infection site. Did the patient have pneumonia and then that proceeded to a bloodstream infection? Did they have a UTI and then they ended up with urosepsis? Do they have an intra-abdominal infection? And then source control methods such as abscess drainage or removal of an infected device, an infected line or catheter, can be done to eliminate the source of the infection. So once you know where that source of the infection is, you want to get rid of that pocket of organisms, because you don't want to keep reseeding the bloodstream with the pathogen while you're at the same time trying to treat it with antibiotics. So very important to try to figure out where this infection is coming from and take care of it. at the source. Also, step 8 is clinical monitoring and supportive care. So during all of this time that we're trying to figure out what is causing this patient's infection, we're also closely monitoring the patient's status and providing supportive care like fluid resuscitation, vasopressor therapy, oxygen supplementation, organ support as needed to maintain and stabilize their hemodynamic parameters and keep those organs functioning while we're trying to help them over the hump and help them recover from this infection. Along with this goes definitive treatment and antibiotic de-escalation. Once the results are available, then the antibiotic therapy can be tailored to the specific pathogen and can be adjusted based on the susceptibility testing. So what we want to do is we want to optimize the efficacy of the treatment, but also minimize the risk of antimicrobial resistance. And we want to try to preserve our armory of antibiotics so that we can use them longer. And so if there is a narrower spectrum antibiotic that we can switch to and get them off a broad spectrum drug, then we definitely want to do that. Also, too, we have to consider adverse effects. Some of the best antibiotics that are going to be the most effective are also really hard on the patient. And so if we can use a different antibiotic that's just as effective for this pathogen, but perhaps with fewer adverse side effects, we want to do that. So de-escalation can be considered if their clinical condition is improving and targeted therapy is available and possible. Then now we finally are getting to the end of the journey here. And so we have follow-up and continuing care. So following the initial treatment, patients require close follow-up to monitor for clinical improvement, resolving the infection, and potential complications. Appropriate antibiotic therapy and duration, and then monitoring for recurrence, because we certainly don't want to release them from the hospital and have them have a relapse. Also, too, many times when they're in the hospital, they're being treated with IV antibiotics. And so if we can transition them to something they can take orally, then the sooner that they can get home and finish their recovery there, which is the best thing for the patient. So now, if I haven't thoroughly scared you by talking about these bloodstream infections and what they involve, I want to go ahead and talk about the yeast bloodstream, the Liaison-plex yeast blood culture assay from Diasorin. And so before we talk about the test itself, I've been talking about bloodstream infections in general, but there are key fungal pathogens that can cause bloodstream infection. And these are the really big and bad ones. So Candida albicans is the dominant species that causes candidemia. And the incidence varies from 40 to 70% depending on the location and the patient population. Candida glabrata accounts for about 27% of all Candida bloodstream infections, but it's also associated with the poorest outcome. And it can rapidly require resistance to azoles, which is one of the classes of antifungal drugs that we've been talking about today. Of course, as you heard in the last talk, Candida auris is associated with hospital-acquired infections, and it's considered a serious global health threat. It's recognized as a cause of fungal bloodstream infections, and of course noted for its ability to persist on surfaces and resistance to multiple antifungal drugs. So this is a real big problem, or can be a real big problem, in our most vulnerable patients who are hospitalized in intensive care or in long-term healthcare settings. So luckily, we have a test for that. The Liaison-plex yeast blood culture assay, or BCY assay, is a test that's recently been cleared by the FDA for diagnostic use, and it's going to be available very soon. Oh, sorry, I got lost control, and now I'm taking back control. Here we go. Okay, so the assay is a qualitative nucleic acid test that's used on our Liaison-plex system to simultaneously detect and identify multiple potentially pathogenic fungal organisms from a positive blood culture. So once that blood culture flags positive, the first thing that you do is a gram stain. And if you see that there is a yeast pathogen in that blood culture, then you can run it on the BCY assay. You can see here that we cover 14 targets that cover 16 species. Most of the important Candida, the ones certainly that we just talked about, but also a bunch of others that may be less common, but if they're causing a bloodstream infection, they're definitely serious enough that we want to know, and we want to know quickly. and also our Cryptococcus neoformans, which I wanted to mention before I lost my place in my slides. Cryptococcus neoformans, or Cryptococcus gattii, very closely related species, which are a significant opportunistic pathogen, and it causes disease, particularly in individuals with weak immune systems. It's the most common cause of cryptococcal meningitis, but can also cause pulmonary infections, skin lesions, and other manifestations. Cryptococcal meningitis affects about 149,000 people each year, and 147,000 of those result in death, or about a 76% mortality rate. And so you can see we are covering crypto as well as these other species. So briefly, let me tell you about the liaison-plex system. This is it's shown here. It is a sample to answer workflow system. It's approximately 1 minute of hands-on time. It has a feature called flex testing. So for particular assays, you can view and pay only for the targets that are of interest. Of course, with the bloodstream infection assays, including the BCY assay, we want to give you all the results because you need to know. But you are sort of selecting which class of organisms you're testing for based on that gram stain result. It's random access, so you can run any test in any of these up to 6 modules at a time. It's scalable, so you can have up to 6 in one instrument. It has a touch screen, so it's really easy to set up and run. It's got a bidirectional LIS to reduce transcription errors and improve efficiency. It's got about a 2-hour turnaround time. And so you get those fast results and prompt treatment and prompt information to the healthcare provider and your antimicrobial stewardship program. And it's very easy to use, considered a CLIA moderate complexity. So here's the workflow. First, the sample's transferred to the cartridge. The run is set up on the instrument. The cartridge is scanned to give the sample ID and the cartridge ID and link those together. The cartridge is loaded, the run is initiated, and then the results can be reported. So I also want to talk to you a little bit about the performance of the blood culture yeast assay. And so this is our positive % agreement for prospective and pre-selected specimens. As you can see here, you don't get a lot of positive yeast blood cultures, particularly if you're doing prospective collection, because these infections, although they're really horrible, they're not quite as common. So in addition, we also do pre-selected specimens that were already previously identified as positive. But in this case, these are blinded and de-identified, and they're randomized and also tested by our clinical sites. So you can see for all of the organisms that we had prospective and or pre-selected specimens, we had 100% positive agreement. And this is compared to the culture methods that were in use at the clinical sites that did the testing. Negative percent agreement was also very high, roughly 98% or higher. There were just a few missed results compared to culture. We had a few false positives. But in each case, if we get a discordant result, we do further testing, and we are able to say that with another molecular test that is FDA cleared, these were also positive, even though they were not positive in culture. So now I want to move on to the contrived. So as I said, since it's really hard to get enough positive samples, it's difficult to evaluate the true performance of the test unless you can test more. And so in this case, we supplemented the prospective and pre-selected sample sets with contrived specimens. And in this case, these were prepared, they were blinded and randomized and sent to the clinical test sites for testing. And whether, we had roughly 49 or 50 positive contrived specimens for each of the targets, and we had 100% detection in each case. Negative percent agreement, also very high. There may be, because this is such a highly sensitive test, you may have 1 or 2 that maybe were false positive, but we don't really expect that to occur very often. And if you have 1, out of 777, that's not really something to worry about. So again, very high sensitivity and extremely good specificity for this test. Now, to finish up, I just want to just briefly touch on a few of the analytical studies that we did, because these are the ones I think are of most interest to you. So we did a limit of detection. We used 2 strains of each of the targets, and we found that the LODs range from about 7.77 times 10 to the 2 to 2.85 times 10 to the 5th CFUs per mil. For growth and detection, we inoculated 3 bottles for each target, grew those, and tested right when they rang positive, and then also at 8 hours after growth. And we had 100% detection at average concentrations of 4.37 times 10 to the 5th to 1.29 times 10 to the 8th. And then blood culture media compatibility, because this is important depending on what blood culture system you're using in your lab. We tested 13 different blood culture bottle types with the BacT-Alert, the BD BacTech, and the VersaTrek, and we found all of them to be compatible with the Liaison-Plex BCY assay. Inclusivity. For inclusivity, we tested 5 strains of each target at concentrations of about 10 to the 5th to 10 to the 7th and had 100% detection. For cross-reactivity, we tested 40 off-panel bacteria and 37 off-panel yeast. And we had no cross-reactivity with the exception of a Candida pseudohamulonii, which cross-reacted with Candida hamulonii and Duo Bush hamulonii at concentrations of 10 to the 2nd and above. Now, these are highly genetically similar organisms, so it's not really surprising that we would have that kind of cross-reactivity. They're thought to evolve from the same progenitor. But again, these are pretty rare, and if it's a pseudohamulonii instead of the hamulonii, it's not really going to impact how we're treating the patient. The Yarrowia deformans demonstrated cross-reactivity with Candida auris at concentrations of 10 to the 4th CFUs per mil. But the Yarrowia is actually the telomorphic phase or the spore-forming phase of the Candida auris, which is the anamorph. And so this is totally expected. We definitely would expect cross-reactivity. Not sure that you're going to see the spore-forming form of this organism in a blood culture, though. And then finally, for interference testing, we tested 18 substances representing contaminants that could be present at blood collection or within a sample. And the only interference that we found was with fluconazole at a concentration of 25 milligrams per liter, which resulted in a false negative call for C. albicans. But if you remember back on our patient journey, we're drawing the blood for culture before we're starting to treat that patient. So unless they had been previously treated and had this drug in their system at this concentration at the time that the blood was called, I don't think we really expect to see that. And so with that, I think I have taken you on a whirlwind tour of yeast bloodstream infections and also the performance of our Liaison-Plex blood culture yeast assay, which will be available very soon. And I'd like to thank you for your attention today, and then entertain questions. You can ask questions either of Dr. Elliott or of myself.
Thank you, Sarah and Sherry, for your informative presentations. Let's now move on to the Q&A portion. Remember, you can submit your questions below in the live Q&A submission box. For our speakers, we will get to as many as we have time for. I believe we have time for just a couple of questions, so I'll go ahead and ask our first one. This first question asks, how common is misidentification and what is it misidentified as?
So I'm going to assume that's for Candida auris, since I did talk quite a bit about misidentification being possible with biochemical and in particular in phenotypic methods. So as far as the frequency of misidentification, this will vary depending on the study that's looking at this phenomenon, but especially with the biochemical misidentifications with the various different platforms. Some studies have found that when they screened a panel of Candida auris, it is 100% misidentified as different organisms. I will say that the organisms that will be that will misidentify as Candida auris can vary depending on the biochemical platform in question, but some of the most common misidentifications are as Candida hamulonii, and then some other closely related Candida species like Candida fumata, Guillemondii, and even some other closely related yeast species like some Saccharomyces or some Rhodotorula as well is fairly common.
Excellent.
I hope that answered your question. Okay.
I believe so, yes. And then our next question here that we're going to squeeze in asks, what gene targets are used to discriminate between Candida auris and other Candida species?
That is a great question. So I can tell you that the Simplexa Candida auris direct assay utilizes the ITS2 region, and this is fairly common for PCR-based methods. It's a highly conserved region within the genome that can discriminate between different, even closely related species of Candida, but is still inclusive enough to be able to detect all of the different clades of Candida auris.
Great, thank you. And I see we do have questions coming in. Unfortunately, this is all the time that we have for Q&A today. So I did want to say thank you to both of you and to ask, did either of you have any final comments you wanted to share with the audience today before we go?
For me, I just want to say thank you for your attention, and we look forward to following up with you if there are any additional questions after we get off the presentation.
Great.
Yes, exactly. Thank you for your time and attention, and feel free to follow up with us with any additional questions you may have.
Wonderful. Thank you again to our speakers for your time today and your important research. Before we go, I'd like to thank the audience for joining us today and for our sponsor, Diasorin. Questions that we did not have time for and any questions submitted during the on period will be addressed by our speakers via the contact information you provided at the time of registration. This webcast can be viewed on demand for 1 year until September 5th, 2025. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, take care, everyone. Goodbye.